Am I audible now? Clearly? Okay, I'm telling you again. Okay, see, triphosphate is going to give energy. Okay, so that is why here we are using deoxynucleoside triphosphate. Okay, so why? Because it's going to give the enough energy needed for formation of new bonds when the phosphate bond breaks. So how in ATP to ADP hydrolysis, that is when the energy is released. Same way here, when the bond is formed, the new bond is formed. For that, the energy is given by the bond breakage of phosphate with it. Okay, so that is how this works. And the next step now. Voice is breaking again, Sushri. Please ask. Sorry? Will No, see, that is what this is going to help in attachment with the complementary strands. So there should be some enzyme that is present, right, for attachment. So this is all a very simple chemical process only. It is catalyzing that reaction. That is all. Even if you don't use it, it is still going to go and bind. But it occurs much slower and might take more energy and time. So to speed up the process, we are using polymerases. That is all. Sushri. Okay. Right. So next step is formation of RDNA. Okay, so what is the step number? 4 or 5? 5, I guess. Okay. So 5, which is going to be okay, thanks. So next is going to be the integration of desirable gene. Now you have a billion copies of this desirable gene. So integration of vector and the desirable gene, which is gene of interest, and when two different genetic material are combined, so you are going to have a vector and then you are going to combine it with your gene of interest. So what you are going to get as a result is what is an rDNA, a recombinant DNA. So this is going to be rDNA, and as Raghu mentioned, it is also chimeric DNA. Okay, so see, normally only the word chimera means it is a combination of many different things. Okay, so now here this is also going to be our rDNA. That is all. Now, how do we do this? We've already studied about this process, if you remember. So, vector and gene of interest, how they are going to be attached to each other is, so both we already know both are cut with the same restriction enzyme. So it is with the same restriction enzyme. If you remember way long back, we have cut this gene of interest from our DNA. The second step, DNA fragmentation. So here we have to make sure that this restriction enzyme that we are using is actually going to be a cloning site on the vector that we are using. So both are cut with same restriction enzymes for the reason that both are going to have complementary sticky ends, so that they simply attach with each other. Okay, so now the thing here you have to remember is, we have already discussed this. I hope you remember it. Now here, if I'm cutting it in this place alone, let us say this is where my cloning site for ECOR1 is present, and I'm cutting it here. How many strands am I going to get? How many strands of vector will I get? How two strands? There is a circle, you cut it at one point, there is just one strand. So it just simply becomes linear. Okay. Okay, so now this is the point where we have cut, and this is where your, I think there is a network issue, students. Okay, so just I will, yeah, there is some network issue. I tried switching between networks, but it's still not working. But anyway, I'll post the video, right? You can check there, or if you have any doubts here, you can ask. Okay, so now we will finish this lesson, or at least try and finish this. Okay, so now here we are adding our gene of interest. Huh. Okay, so now this is where the cut is going to be made, right? So if a circular structure is going to be cut just at one point, it just simply gets torn there and it becomes linear. Okay, so this circular vector is going to become linear if you are cutting it at one point. Now, if you are cutting it at 2 points, you will get two strands. If you are cutting it at 3 points, you will get three strands. Okay. So, these kind of questions they may ask, but we know that in an ideal plasmid, there should be only one cloning site for one restriction enzyme, so it becomes linear. Okay, so and then we already have our gene of interest, which is also going to be having the complementary strands. So we are going to attach our gene of interest here, and then what happens here, it is going to form our recombinant DNA. Okay, so ligases will be used in this case. I'm sorry. So this is going to be rDNA that has been formed with the help of restriction enzymes and ligases. Okay, so now a recombinant DNA has been formed. So the next step, we know that we are going to put this into the host organism. How are we going to integrate it or enter it into the host organism is our next step. Did you all understand this part, integration of vector? This is the whole half of the lesson. Okay, so where we are going to cut the vector and the gene of interest with the same genetic material, sorry, same recombinant, same restriction enzyme, and then that gives us the rDNA. Okay, so now if this linear DNA, please pay attention, if this linear DNA is going to be cut at two different regions, there you end up with three strands. But if you are cutting a circular DNA at two regions, you are ending up with how many strands only? Two strands only. The number of cut is going to be the same. In the linear DNA also you have cut it at two regions only. In the circular DNA also you have cut it at two regions only. Okay, so but the number of fragments you will be getting is different. So please keep this in your mind. Okay, right. So if you have any questions, please ask. No doubts. Okay, then we'll move on to the next part of this. Okay, so the next step is introduction of the rDNA into a competent host. So now again, I think we have already seen what a competent host is going to be. A competent host is something that can take up the DNA, it can take up the recombinant DNA, and it can express that character. Putting it in an organism that cannot express it in any way is not going to be ideal. So a competent host. So now how we are going to make that host competent is what we are going to see. So now before that, one thing you should know is DNA is an hydrophilic molecule. What do I mean by that? Hydrophilic? Yes. If it is water loving, then what would it hate? It is hydrophilic and lipophobic. Okay. So, yes, Anto. No, your hand was raised, so that is why. Okay, fine. Okay, so no problem. Yeah, so the DNA is going to be hydrophilic and lipophobic, which means what? If this is going to be your cell, this is your host cell. Let us say this is going to be E. coli only, just for example. So now this is where your gene of interest and your rDNA has been made. You have made this recombinant DNA. Now this has to go inside. Now normally DNA is going to be lipophobic. Now even if you find a way to remove its cell wall, it is not going to cross its cell membrane. Why? Because this DNA is hydrophilic and lipophobic. So now you have to make sure that the DNA and the cell membrane of your host cell are attaching with each other. So how we are going to do is, that is how we are going to make transformation by treating it with one minute.